Hello and welcome to Samish Bay. I'm Jen and this is Stanley. Samish Bay is located about halfway in between Bellingham and Anacortes. If you drew a line between the two on a map, we would be right about in the middle of that line. Samish Bay is fed by the Samish River, as well as a few other smaller creeks and sloughs, like the Edison Slough and Oyster Creek. On a field trip to the Mudflats, your class would typically split into groups. Each group would lay out a transect stretching from the rack line, either down to the edge of the water or down about 50 meters. You would then collect data from 4 quadrats evenly spaced along the transect. When you lay out quadrats, make sure to lay each one out in the same orientation relative to the transect, so that you're not selectively measuring anything. When I do it, I like to lay the quadrat parallel to the transect line, with the top left corner at the measurement mark. At each quadrat, count the number of individuals of each species present. Select 2 to 4 individuals of each species and transfer to a Ziploc bag or biota jar. Make sure you label your specimen with the date, site and quadrat information, your group number, and your class. Back at the lab, you will clean off any sand clinging to your specimens and dry them at 60 degrees Celsius. This drives out most water while minimizing the loss of volatile organic compounds. After drying, the specimens are weighed. From this number, you can calculate the biomass of each species for the area surveyed. Specimens are then burned in the ash furnace to remove all organic matter and weighed again. This number provides the mineral content of the biomass. These tidelands are host to an enormous amount of primary productivity. 2 species of eelgrass abound here, both the broad-bladed meter-long native Zostra marina and fine-bladed introduced Zostra japonica. Zostra marina is especially important because it provides habitat for juvenile salmon and a myriad of other species. You should also find ulva species sea lettuce and may find a few other marine macrophytes. There is also a very dense benthic macroinvertebrate community living in these mudflats. Each of these holes marks a sandworm, clam, or arthropod living in the mud. While it can take experience to tell the difference between a butterclam hole and an arthropod hole, horse clam and softshell clam holes are both larger and round. Sometimes, but not always, you will see a siphon protruding from a clam hole. Even if the siphon is not protruding, you should be able to feel it if you stick your finger down the hole. The clam, correctly sensing a predator, will retract its siphon down the hole and into its body. A wormhole will have a cleaner, sometimes even funnel-shaped entrance. If you stick your finger down a wormhole, you will not encounter any objects or resistance. If you scoop out some mud with a shovel, you can see exactly how thin the oxygenated layer is by where the mud goes from brown to black. Long siphons and deep burrows allow clams and other animals to live deep under the oxygenated layer, where predators cannot eat them, just stretching their siphons up to feed and breed. Another species that you are likely to find in Samish Bay is the invasive bamboo worm. They are native to the Atlantic and Gulf coasts and first arrived here about 15 years ago. These annelids are extremely fragile and will often break if you try to dig them out of the mud for a biomass sample. Bamboo worms have burrows with small openings only a few millimeters across. These worms construct tubes to live in about 20 centimeters long, perpendicular to the surface, and sometimes you can see the tubes sticking up above the surface of the mud. These worms form patches of dense colonies that cause the mudflats to become porous and spongy. People commonly get stuck while walking through patches of bamboo worms. Since I have a vile sense of humor and believe in the power of experiential learning, I typically do not tell students about bamboo worms until at least a few people are simultaneously stuck. Field trips to the mudflats also involve water sampling. A group will take dissolved oxygen and pH readings, as well as taking a water sample from the surface. Remember to record dissolved oxygen in both percent and milligrams per liter. Normally, you would take conductivity using the same meter you use to record pH, but the conductivity of seawater is so high it is over the range of our handheld meters. When you take your water sample, be careful not to scoop up mud stirred up by your boots. Another group will take a plankton tow. Since you're on the shore instead of in a boat, this plankton tow will be done cowboy style. Just set up your tow, strip out enough line, spin overhead, and release like a lasso. Tow the net back slowly at about the same rate you would perform a plankton tow by boat. If necessary, you may repeat the process. The plankton tow may be preserved with Lugol solution or checked immediately with a Sedgwick raptor cell microscope slide in a compound microscope. Another group will be responsible for taking salinity measurements. To do this, you will use a refractometer. Before going out in the field, check calibration of the meter. If necessary, adjust the set screw so that it reads 0 with deionized water. To measure a water sample, fold back the plastic cover plate and ensure that both the plastic part and glass face are completely clean. Holding the glass face level, take a dropper pipette and place several drops of seawater on the face. Gently fold the plastic plate back in place, making sure there are no air bubbles. If you get any air bubbles, start over. Hold the meter up to your eye, facing a good light source. Read the line where the blue and white parts meet. That's your salinity. Our meters read in both percent and specific gravity. I recommend recording both numbers. When you're finished, rinse the business end of the refractometer with fresh water and dry before returning to the case. If you come to Samish Bay, you will notice something unusual about my community. It's full of shellfish farms. Most clams farmed here are the Ruditapes philippinorum manila clam. We're here at Commercial Clam Farm in Samish Bay. If you look behind me, you see lots of dark strips on the ground. Each strip is a row of clams that have been planted. Clams are planted when they're 1 or 2 millimeters in size, and the farmer will broadcast them out like corn seed. And then once they're planted, the farmer will cover them with plastic netting to exclude predators like birds, crabs, moon snails. Once they're about 1 inch or 2 in size, the farmer will peel back the netting and then either harvest them by hand with a clam fork, or else they'll come through with a tractor and use a special implement similar to a tulip bulb harvester, like the ones they use out in the Skagit Flats. The main oyster grown in Samish Bay is the Pacific oyster, Crassostrea gigas. Here you see 2 different methods of oyster culture side by side. Up in these bags, that are suspended from ropes. You see Pacific oysters grown in the Shinoku method. And when the tide comes in, it floats these buoys up, and it causes the oysters to tumble around in the bag. This breaks all the little edges off of the shells and creates a beautiful, smooth, rounded oyster, kind of similar in appearance to a horse's hoof. And that's highly prized for raw consumption because it has a deep cup and a nice little solid meat. Down below, there's a traditional culture method, and these are a little oversized. They're older oysters. Oop, I just poked myself on a sea urchin. And this is a cluster culture method. And when these are grown, there's always an empty shell on it. And that empty shell is called a mother shell. It's put into a vat of water with larval baby oysters called spat. And the spat will settle onto the mother shells. Then the farmer will buy these shells with little baby oysters stuck to them, and they'll scatter them out on the oyster beds until the oysters have grown large enough to eat. Ooh, I have a crab on me. Once they're ready to harvest, the farmer will bring them in, and they'll take a crude metal implement and break them apart. In large farms, they'll even use a rotating drum to break these individual oysters apart, so each one can be sold. In 1889, when Washington became a state, terrestrial land belonging to Native Americans was not the only property sold to settlers under the Homestead Act. Tidelands adjoining terrestrial properties were also sold. In 1895, these tideland sales were expanded to promote oyster farming and stimulate the economy. By the time the Gisborne Amendment forbade sale of state-owned aquatic lands in 1971, our state had sold about 70% of its tidelands, about 30% of which are in the Puget Sound. Washington is one of the only states in the USA to allow private ownership of tidelands. Interestingly, while the Bolt decision of 1974 affirmed the rights of Washington tribes to act as co-managers of natural resources and collect 50% of all fishing harvest, this was not recognized to extend to shellfish harvest until the Rafiti decision of 1994. This led to some colorful confrontations in densely populated areas with privately owned tidelands, at least until a 2007 court decision awarded tribes $33 million to buy and lease tidelands for their exclusive use. Regardless, if you decide to walk on a beach in Washington state, you should check first to see if it is private or public, and be respectful of the neighbors. While the history of Washington shellfish farming is an interesting anomaly for our nation, shellfish also provide important ecosystem services by filtering the water. One adult oyster can filter about 50 gallons or 190 liters of water per day. Eutrophication can cause an ecological worst-case scenario in these kinds of waters. If you have enough eutrophication to cause low oxygen dead zones, filter feeders can die. Without filter feeders, the excess phytoplankton is not removed from the water, and the ecosystem is hit even harder. A walk through an oyster bed will also show the habitat niches that they provide to a variety of species, including small fish, juvenile crabs, and other invertebrates. While oyster beds aren't quite as favored as eelgrass by salmon fry, studies have shown that oyster beds in Samish Bay provide similar habitat. Thank you for joining me at my home in Samish Bay. Stay healthy and safe, everybody. Bye!